when it comes to binding to things, I guess. So without any, I don't know if these are agonists or antagonists at each of these sites, whether the binding of DMT affects these sites in a positive way or a negative way, but it does have a binding, or it does have binding effects at all of these things. A few dopamine, or a couple dopamine receptors, histamine, norepinephrine, basically like all the 5-HTs basically. Sigma 1 and 2 I'll be talking about. And the transporters, dopamine transporters, the serotonin transporter, which is on the outside of the cell, I believe, and transports serotonin and probably DMT into the cell. And then the vascular monoamine transporter 2 is what transports the serotonin and maybe the DMT. Nobody's done the test. Well, there's some, I won't talk about it because it's all very unpublished right now. Into the vesicles, which will then be released into the synapse on response. This video down here, Nick Cozy is part of a team that found out that dimethyltryptamine was an endogenous ligand to the sigma receptors, which hadn't been found beforehand. It's been an orphan receptor for a long time. So I'm going to talk briefly about the 5-hydroxytryptamine receptors, the serotonin receptors, as well as the sigma receptors. So they're all G-protein coupled receptors. 7-membered helices, transmembrane helices. You have your binding site, ligand binding site up top. And then you have this G-protein, which once the ligand binds, it detaches and goes and does other things within the cell. And I'll be highlighting 2 mechanisms that the serotonin, or that the various serotonin receptors work on. So the serotonin systems are like, serotonin neurons are mainly centralized within the raphe nuclei in the midbrain, but they project all the way throughout the brain, or into the cortex and around. It's sort of thought that serotonin acts as a wakefulness sort of thing. So you wake up, and you might be groggy, you might not, whatever, get a burst of serotonin, and it'll start to wake the rest of your brain up. Serotonin usually acts, in that sense, as an excitatory neuron. Yeah, it should be noted that serotonin is involved in, I guess, stress-induced responses. It'll elevated serotonin will elevate adrenocortical hormones and prolactin, vasopressin, oxytocin, which means that it interacts with the hypothalamic pituitary adrenal axis, the adrenal glands located above your kidneys, and produce epinephrine and norepinephrine, I believe, implicated in fight or flight response. So that's sort of, you know, all revved up and know where to go sort of thing. So again, this goes back to the serotonin being a very primal sort of molecule. It was originally isolated from the gastrointestinal tract along, or same as GABA, glutamate, and acetylcholine. So this is a study that was done in '94. It compared the amino acid sequences of all these molecules. It's kind of okay. It's an okay study. I don't know how good it is. It's okay. But there's so much diversity in the G-protein receptors that it's hard to do this. But you can see, so what we have here is millions of years ago, so around 800 million years ago, you have sort of the first differentiation from the primordial 5-HT receptor into the 5-HT6, which goes all the way a straight line, the 5-HT2, and the 5-HT1, which can be split into the 5-HT5, 7, 4, yes. And 3 is not on there because 3 isn't a G protein coupled receptor. It is in fact an ionotropic receptor. It gates an ion channel, which is very interesting because, again, serotonin having all these different effects on so many different types of receptors. And at the top there, the color coding is just sort of the different G proteins that are attached to the various receptors. And we'll go into that. You can't read it here because I forgot to change the color. But this study actually suggested that muscarinic, dopaminergic, alpha adrenergic and beta adrenergic receptors are more homologous to the 5-HT1 type than they are to the 5-HT2 and 6. So he suggests that all these receptors actually evolved from a primordial 5-HT1 receptor, which again adds strength to the idea that serotonin is a very, very primordial system. So this is a great diagram. It sort of shows you the groupings of how the various G proteins that are coupled to each of the different 5-HT receptors interact with the cell at the cellular level. Two basic systems, like we'll exclude the ligand-gated ion channel, the 5-HT3, and we'll just focus on the cyclic AMP pathway and the, I forget the name, polylipase C. Anyone? Oh, here we go. Phospholipase C. Yeah. So the 5-HT1s have a negative effect on the cyclic AMP system. The 5-HT4, 6, 7, and 5 have a positive effect using G protein I/O and G protein S respectively. And the 5-HT2 often implicated, or the 5-HT2C at least, is often implicated in hallucinogenic effects, is responsible for the phospholipase C pathway. The things with stars beside it are ones that DMT binds to, and the ones with the cross are ones that have actually shown agonistic action. But a lot of these haven't had agonist-antagonist studies done on them, so. But the binding profile is rather impressive. So I'm gonna focus on the 5-HT1 and the 5-HT2C and A receptors to illustrate these pathways. So first, the cyclic AMP pathway and the GIO G protein. So when you have your DMT, it binds here. The G protein will be released, and the gamma beta subunit closes the potassium channel, and the alpha subunit travels over here and inhibits adenylene cyclase, which reduces the amount of cyclic AMP in the cell and lowers various kinase activities and basically reduces the activity within the cell. It also has some effects on closing calcium channels. The concentration of calcium is very important in determining the overall activity of the cell. Opening of calcium can trigger cells to release neurotransmitters, regulates muscle contraction, and very high concentrations of calcium trigger cell death. So calcium signaling within the cell is very important, and dimethyltryptamine by various systems that I'm describing is intimately linked to those. So in opposition to this pathway is that of the 5-hydroxytryptamine 4, 6, and 7 pathways. They're coupled to G protein S. So they're all very similar, and their distributions are sort of what makes them unique. Oh yeah, so the 5-HT1s are inhibitory, and the 5-HT 4, 6, and 7 are all excitatory. And again, this is just sort of places where you can find this inhibitory function of the serotonin. Now onto the 5-HT2 and 2A and 2C receptors. They're coupled with G protein Q or it looks like a heterotrophic protein or whatever, GQ-G11, which GQ, it activates phospholipid C, which cleaves this molecule PIP2, phosphatidylinositol 4,5-bisphosphate. It's a molecule sitting within the lipid bilayer surrounding the cell, the outside of the cell. Phospholipase C is embedded in the cell membrane, sorry. And it splits it into diacylglycerol, which is sort of like a fat molecule with 2 fatty acids coming off of it, and inositol triphosphate. And the DAG molecule, the diacylglycerol, will have some effects on activating various kinases and increasing activity that way through metabolic effects. I want you guys to pay close attention to the IP3, the inositol 3-phosphate that came out of that cleavage, because there are inositol 3-phosphate receptors within the endoplasmic reticulum, and the endoplasmic reticulum is on the inside of the cell, and it is actually a large warehouse full of calcium, basically. Well, like as far as we're concerned. Most of the body's calcium is not stored in your bones, it's stored in the endoplasmic reticulum or in the muscles, the similar structure, the sarcoplasmic reticulum. But anyways, so the IP3 will bind to calcium channels on the endoplasmic reticulum and trigger them to open. And raising the calcium levels and exciting the cell to do whatever the cell is going to do. In the case of neural axon, maybe release neurotransmitters. The calcium is doing a similar thing as an action potential would do along the cell, because as the action potential comes along, the sodium and potassium gates open up with the change in voltage, and they rush in and then allow the calcium channels to open, which calcium from the exterior of the cell rush in, raising the calcium concentration and activating the cell. So this is a way that these metabolic pathways, so it's not an ion channel, it's not directly affecting calcium by binding of DMT. The DMT binds, activates the G protein, which activates this lipase, breaks apart a molecule within the cell. So this can be a common pathway shared by many G proteins. And then in that way, and then the IP3 comes, opens up the calcium, and you have again a raise in calcium within the cell, exciting the cell.